Hello, everyone. Thank you for coming to our March online lecture event. We are the Center for Climate Science and Engineering at the University of Toronto. We try to host monthly guest lectures. My name is Mohamed Jalayer. I am the CSE manager. Today, we are pleased to host Dr. Anirban Kundu from Antithesis Consulting. First of all, I'd like to go through the land acknowledgement. We wish to acknowledge this land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still to home to many Indigenous people from across the Turtle Island, and we are grateful to have the opportunity to work on this land. If you are new to get our guest lecture series, welcome. The CSE is a research and education center within the Civil and Mineral Engineering Department at the University of Toronto, and it was established in 2019. The CSE consists of a diverse and multidisciplinary team of faculty members who collaborate on interdisciplinary research at the intersection of climate science and engineering. Our center's director is Ömer Can, and she focuses on structural analysis. Daniel Paulson focuses on life cycle assessment. Ahmed Ibrahim works on hydrologic variability and processes. Karen Smith focuses on atmospheric science. Mohamed Bashir focuses on hydrology and water resource systems. Paul Kushner looks at climate dynamics. Sam Markov from the University of California, Merced focuses on sustainability and resilience. And then me, the CSE manager. So one of the primary initiatives undertaken by the CSE focuses on providing education in climate science and engineering. We have developed and launched 2 graduate level courses at the University of Toronto. Additionally, we have created 8 online learning modules that are now accessible for use across higher education institutions in Ontario. We also design and run 2 courses through University of Toronto's Deep Summer Academy for high school students. Furthermore, our faculty members are engaged in collaborative research projects at the intersection of climate science and engineering. Also, our team organizes outreach activities just like this one, where we invite public and university community to give a presentation on topics that are aligned with the mandate and vision we have at the center. Now, I'd like to introduce today's guest lecture speaker. Dr. Anirban Kundu is a senior sustainability consultant at Antithesis Consulting, where he helps organizations navigate environmental, climate, and socioeconomic challenges while building resilience and sustainability programs. His expertise spans corporate sustainability, conservation agriculture, and climate risk. Before Antithesis, he was a program manager with the Impact Challenge in Climate Innovation team at google.org, where he contributed to assessing AI-driven solutions for climate mitigation and adaptation. Dr. Kundu holds a PhD in environmental engineering from McGill University, where his research focused on integrating environmental microbiology, soil geosciences, and multifunctional nanomaterials to address environmental remediation of diesel contaminated subarctic soils and promote sustainability and sustainable agriculture. He is also accredited in sustainability and climate risk and holds the prior degrees in bioengineering and biochemical engineering. Beyond his professional role, Dr. Kundu manages the editorial committee of the Canadian Science Policy Centre, Canada's largest science policy community, and serves on the advisory board of Waymakers Canada, the nation's sole work integrated learning program for post-secondary students. Additionally, he delivers guest lectures at universities, covering topics such as environmental sustainability, diversity, equity, inclusion, and skill development for future readiness. He is going to give a presentation on multifunctional nanomaterials for agriculture resilience and progress towards sustainable development goals too. Dr. Guntu, welcome. We are very happy to have you here today. Now the floor is yours.
Can you confirm if you can see my screen?
Yeah, I can see the screen.
Brilliant. Okay, I will just make it full screen and perfect. Okay,
it's great now.
Perfect. Thank you very much. It's great to be here. Thank you for allowing me to talk about how we're using multifunctional nanomaterials for agriculture resilience and towards progress towards SDG 2. I'm going to start right into the topic, which is the need for agricultural resilience systems. So if we look at the latest, so if we go very broad level, we look at the Sustainable Development Goals. One of the most important goals is food, ensuring there is enough food for the world, right? And if we look at the latest United Nations figures, we will see that by 2050, we need to feed an additional 10 billion mouths, right? Now, further analysis tells that right now, 10% of the people worldwide suffer from hunger. 1 in 3 people face food insecurity. And there is, in general, a lot of food wastage, right? So global food systems in general face adversities due to conflicts, due to climate shocks, due to social inequalities, and all this hinder progress towards ensuring there is enough food, resilient, safe food for people in the world. And that is one of the cornerstones of UN SDG 2. In addition, the pandemic led to more people facing hunger in 2021 compared to 2019. So these figures emphasize the need for resilient agricultural systems. In addition, these figures also emphasize the need for agricultural systems that can cope up with climate risks, that can cope up with the increasing use of fertilizers, and make sure that we are controlling the environmental impact of food systems, because agriculture is also one of the major emitters of GHG emissions. So food needs to be resilient. Food systems need to be resilient. They need to have a sufficient protection against climate risks, and they also need to be delivered and implemented in a sustainable and inclusive manner. Now, if I look at the climate smart agricultural landscape, when I mean climate smart agriculture, I mean agricultural systems that are protected or that have mechanisms, robustness against climate risks and environmental risks. There are a number of climate smart agricultural technologies and strategies that are available today. A lot of them are in application. There are some technologies which are more in the research phase. And what I've tried to do here is I've tried to paint an overall picture of these technologies. So starting with indoor agriculture, and before I proceed, one of the major governing factors behind climate smart agriculture is to make sure agriculture uses less, it leads to less environmental impact, it leads to greater yield, crops are more protected against climate risks, and it can lead to a system that can sustainably feed the growing population, right? So if I look at indoor agriculture, there are a number of techniques that are available now, like hydroponics, aeroponics, and vertical farming, which remove the reliance of agriculture on soil, but instead look into model systems involving nutrient solutions or spraying of roots with nutrients or growing crops in a stack-wise manner. The reason behind, or the inspiration of these systems is that there is less reliance on land. So in vertical farming, if you look how plants are grown in a stack-wise manner, you don't need a lot of land. You need more stackable systems to grow these crops, right? You also eliminate the large-scale use of agrochemicals and pesticides, and generally, because these crops are grown in a controlled environment, they're generally protected against climate extremes. You can also have more enhanced yields over time. For precision agriculture, the whole inspiration behind precision agriculture is to leverage data, to leverage technology using various data acquisition methods like drones and sensors to collect data on crop health, ecosystem health, environmental health. And you analyze this data, and real time you can understand where you need certain protections or certain interventions to ensure that the food supply is not threatened. So you could have a continuous data monitoring of certain climate risks over time, or monitoring crop growth and health parameters. And this multi-parameter modeling can help drive a data-driven approach and protect against abiotic and biotic risks. Similar concept is smart irrigation, which is adjusting the soil watering based on certain sensors and weather and climate data. And smart irrigation is having a lot of impact because right now there are a lot of agricultural lands that overlap with water-stressed lands, or groundwater is quite stressed, which means there is not enough water for irrigation purposes. So really identifying where irrigation is needed and at what amounts is very important to control the wastage of water. Then we have more advanced technologies or emerging technologies like bee vectoring, which is bee-assisted precise delivery of biocontrol agents. So bees, they hop from plant to plant, and that can be utilized to deliver biocontrol agents specifically to the plants. And this has been interestingly applied to a wide range of crops like fruit, strawberries, apples, various fruits, and some reports have reported more 30% increased in yields. But again, this is a more emerging technology. It's being tested and tried in field trials, and we need more such field trials to actually understand the true effectiveness of bee vectoring. You have modern biotechnology and omics-based approaches where you go deep into using omics tools, genomics, transcriptomics-based tools to precisely manipulate the crop genomes. So you identify if there are certain gene-based factors that lead to vulnerability against certain risks due to climate or due to pests, and then you either silence those genes or you perform genetic manipulations to develop strains that are better protected. For example, you could have drought-resistant strains, or you could have pest-resistant strains, and so on. And lastly, the focus of today's presentation is nanotechnology, using nanoscale materials. And as we go forward, a lot of the things that we've just discussed about controlled nutrient delivery or omics-based approaches for precise manipulation of genomes, these can be and have been applied using nanoscale materials for a number of different purposes, for targeted fertilizer delivery or for enhancing seed stress tolerance and shelf life. And going forward, we will dive deep into some of these applications. So the focus of today is to understand why nanotechnology, what is in this field that makes it such a promising platform for agriculture. And I always start by giving this definition. It's more or less a widely accepted definition of nanomaterials, which is materials that are manufactured at or within the nanoscale. So any dimension of the material has to be within 1 to 100 nanometers. Now, for the purpose of the applications that we will study today and going forward, usually there are a number of properties of these materials that make them very attractive for particular applications, more so for agriculture, and of course for other applications like biomedicine. The first one is multifunctionality. So nanomaterials, they are very small in size compared to bulk materials, which gives us the ability to combine various physicochemical properties, electrical, magnetic, optoelectronic, and so on, to enable diverse applications, right? And for each of these descriptions, I have provided a guiding question that is relevant to agricultural application. So in this case, multifunctionality, the guiding question would be, how can we identify these physicochemical characteristics and thereby design materials that have multiple functions? And these materials have been developed. So you could have a material that not only provides agrochemicals in a precise manner to the crop, but also leads to drought resistance and also leads to certain disease tolerance. So there's one material whose properties have been tuned and tailored to provide multiple end benefits to the plant. The next one is selectivity. So nanomaterials, from a synthesis and chemistry standpoint, they can be surface functionalized, or they can be, certain properties can be attached to the nanomaterials and program them to preferentially interact with certain targets, particular targets. So they interact with these targets, they bind to a particular target or zone, and they release their cargo or active ingredient. And the guiding question here would be, how can we enable nanocarriers to supply therapeutic agents or biocontrol agents to plants? The third one is tunability. How can we tune or engineer reaction conditions? So this is going deep into the synthesis of these nanomaterials. Nanomaterials are synthesized using various reagents and temperature and time and pH conditions of the reaction. So how can we engineer these conditions to modify the multifunctionality of nanomaterials, right? So how can reaction chemistries help us prepare nanoparticles with different physicochemical properties that can impart multifunctionality? And the last thing is scalability. If you have a technology that works in the lab scale, it is very essential to scale that up and find how it works and in the field scale, and especially that's relevant for agriculture. So how can we, based on the reaction conditions of the lab scale them up maintain productivity, maintain yield, and physicochemical characteristics, consistent quality, both at the lab scale and at the field scale. And in this case, it's very important because in agriculture, effectiveness, the one of the sole parameters governing effectiveness is scale. So you would need to prepare these materials and identify the infrastructure that is required for field scale application. And in parallel, what are the training and technical resources that are needed to promote field applications? So considering these four properties, multifunctionality, selectivity, tunability, and scalability, we are going to go deeper into understanding some of the applications of nanotechnology in agriculture, right? So I've started by providing a broad overview of the four main applications that we are going to go through today. And then in subsequent slides, we're going to go deep into each and one of them, one of these applications. We are going to go into what is the mechanism of the application, what plants or crops have been the subject of these applications, and the effectiveness and the research results that have come out of studies on these themes. So the four themes that we're going to look into today are nanopesticides or nanoagrochemicals, nanosensors, and nano-based applications in genetic engineering, and seed resilience. Now, for nanopesticides, the main point that we're trying to look into here is how can these systems supply pesticides and agrochemicals in a more sustainable manner? Now, we all know pesticides are not good for plants and crops. The reason being, there is often wastage of pesticides. For example, you supply 100 units and the plant might need 20 units, but the remaining 80 units is being wasted in the soil, which leads to downstream pollution. So how can we make sure that we supply only what is needed by the plant at the exact location in the plant, right? And that would then not only supply the exact amount needed by the plant, but also prevent wastage to the environment. And the way nanomaterials help us in this venture is they help in targeted attachment and slow localized release of this pesticide or agrochemical or active ingredient in the particular plant location. In case of nanosensors, the point is how can we monitor plant health, right? So, when plants are exposed to climate-related stresses or even wounds, for example, they will release stress molecules. So if we monitor these stress molecules, then we will, over time, we can get the real-time data of understanding, okay, this particular stress molecule is being produced in large amounts, then it can help us monitor the particular shock of the plant to certain stress. And in that way, we can prevent these stresses by taking the appropriate adaptation and resilience methods. So how can we monitor plant molecules to detect stress and resource deficiencies due to climate and environmental factors? The third one about genetic engineering is how can we use nanocarriers that deliver in a high throughput manner biomolecules and nucleic acids that cross the plant cell wall and can enable certain modified characteristics in the plant for example, stress tolerance or for heat resistance and so on. So the mode of action here would be more towards targeted delivery of biological cargoes. This is very similar to the nanopesticides one. The only difference is the pesticides one is more about the agrochemical, whereas this is more about the nucleic acid or the DNA or RNA or protein or whatever we want to, the biomolecule complex that we want to supply to the plant. And then seed resilience. So how can we develop a certain nanomaterial-based protective layers or coverings that can improve germination of seeds, that can enhance pathogen resistance, right? So here there is a lot of work being done on seed coatings that improve seed tolerance, that provide a very enabling environment for the germination of the seed in adverse conditions like drought and so on. So these are the four pillars that we are going to look into today with some detailed examples. So we're going to start with nanopesticides and nanoagrochemicals. And the mechanism there is, imagine you have the active ingredient. So imagine copper, right? Copper or zinc. Copper or zinc is copper, zinc. These are very important micronutrients for plants, right? Now, how can you supply this micronutrient in a targeted manner to the plant without causing any sort of wastage? So one way you could do that is if you can package that inside a nanocarrier porous shell, right? And that porous shell, over time, because it's an ionic complex, the active ingredient in a plant media would dissolve. And due to these pores present, the pores would facilitate the slow release of these copper or zinc ions to the plant. However, if you only had the zinc or the copper in the nascent form, then the release would be much faster, right? So dissolution and due to the presence of the mesoporous shell, the slow release of the ingredient, the release of the ingredient is happening in a much slower and sustained manner as opposed to that in the ionic form. So essentially, you have one entity that is giving these important ions over a long period of time. So as opposed to something that's giving a burst release. So there's a lot of release initially, and then the release just stops. What we've seen both in our lab and in other studies is the slow release can over time lead to can sustain plant growth, and the plant takes up the nutrients. So you apply these nanoparticles in the leaf, but these active ingredients are going to be translocated all throughout the plant over the slow release period of time. And this is one of the mechanisms by which nano agrochemicals and nanopesticides are being applied today. Now, you could have a porous shell, you could have a polymeric particle, but the whole point here is to ensure that your active ingredient is being released in a slow and sustained manner over time. So I've presented two examples from the literature. The first one is a study in our lab where we developed these mesoporous silica nanocarriers that have sustained zinc delivery. So zinc is an important micronutrient to tomato plants. So we grew the tomato plants in the lab in a hydroponic system. And if you look at the figure on the left, the top panel figure on the left, the green the green dots are from the nascent zinc oxide. So we have monitored the dissolved zinc over time, and you see there is a burst release. So within a few hours, the amount of zinc that's released is very high. But if you look at the red, which is basically the zinc packaged in a mesoporous silica shell, the release is much sustained and much slower over time. So the plant gets the zinc over a sustained and long period of time. And on the right, we actually tracked this zinc across the plant. So we dosed these materials in the leaf, which is why you see a lot of green area shaded, which is the concentration of zinc in the leaf. But then in 3 days, the zinc reached to other parts of the plant, which are the other areas of the pie chart. So it reached the stem, the root, and other areas in the plant, which basically told us that zinc in silica nanocarriers were an effective delivery agent of zinc to tomato plants. There is another really amazing study which is done to test slow release of nitrogen for rice. So similar concept, if you look at the figure on the left, the blue bar, the blue curve is urea hydroxyapatite nanoparticles. So urea is an important nitrogen source. And what these authors did is they interfaced the urea in hydroxyapatite and they prepared this complex. And they found that this complex releases nitrogen over an extended period of time, as opposed to only the urea, which, if present in a aqueous media, led to an immediate burst release. What I really like about this study is they did experiments in the field, right? So if you look at the figure on the right, they found that when no fertilizer was applied, the yield was much less compared to when only granular urea was applied. So this is the conventional fertilizer source versus when urea hydroxyapatite nanoparticles were applied. So this sort of tells you that if you have a way of supplying the active ingredient over a period of time to the crop, then over a period of time, the productivity, the yield of the crop increases. And this is important because not only are you having an increased yield of the crop, but you're also making sure you're not wasting enough nutrients. So if I look at the literature, a snapshot of the literature will tell me that there are a number of carrier matrices that are available today to deliver active ingredients. You have mesoporous shells, you have liposomes, polymers. You also have clay nanocomposites, fantastic materials. They have clay sheets, and in between those sheets, the lumen, the hollow area, you can interface the active ingredients. So which is why that's the figure, that the schematic that is represented on the right. And a lot of plants have been subject of nanopesticide and nanoagriculture studies. So potatoes, spinach, rice. I believe this is one of the most investigated areas, and this has enormous potential going forward. So boiling down to the strategic advantages of nanopesticides: controlled and targeted release, versatility in active ingredient. You can go from pesticides to nutrients and hormones. You minimize waste, and there is an ability for you to prepare multi-cargo release carriers. You could have one nanomaterial that releases copper and zinc and other micronutrients. And these studies have been done in the literature. Moving to the next topic of nanosensors, right? So the whole principle being here is to monitor the response to stress compounds. And this can be abiotic stress, it can be physical stress, climate stress, and so on. So usually when plants are faced with different types of abiotic stress, like heat, light, excessive salinity, and so on, plants release stress molecules. Examples could be hydrogen peroxide or salicylic acid, indole acetic acid, ethylene. These stress molecules, if they can be monitored using a nanomaterial, then we can get an idea of what is the impact of the stress on the plants. So what people have done in the literature is they have prepared nanoparticles that respond to the presence of these stresses using a sort of fluorescence or quenching. So so you have a nanoparticle, it has a receptor that on identifying the stress molecule either gives out a fluorescence, or if it already has fluorescence, it is quenched. So you can use that signal to identify when the plant is in stress and when it's not, and you can monitor the stress over time. And this signal is measurable or detectable. Think of near emission NIR radiation, or think of Raman spectroscopy that can help you measure and detect this signal over time. A number of fantastic studies over here, and going to the first one. So black rot is a disease that the authors wanted to monitor in bok choy, right? Now, they prepared this carbon nanotube sensor, which can detect salicylic acid at endogenous concentrations. Now, these stress compounds, they're released at very small, minute amounts, right? So you need really precise detection systems. So salicylic acid is one of the stress compounds that was released in bok choy when due to black rot disease. And what they found out, so they gave this image of a leaf. So on the left side, you have this carbon nanotube sensor that is designed in a way to detect salicylic acid, and on the right, you see the reference sensor. So if you look at the concentration of salicylic acid over time due to infection over time, there is a clear fluorescence that you can see on the left, but you cannot see on the right. This fluorescence is basically telling you that this carbon nanotube sensor can detect salicylic acid due to the infection of bok choy, infection in bok choy. And this can help in early diagnosis of plant-related stresses. So this is a type of non-destructive, real-time detection of stress-induced biochemical signals. In a similar fashion, the next study is doing just the opposite. Instead of fluorescence, is more detecting quenching. So this is the monitoring wound response in spinach due to a single-walled carbon nanotube. A sensor was developed using single-walled carbon nanotube for monitoring wound-induced hydrogen peroxide generation. So in the first case, salicylic acid was the molecule that was being released due to stress. In the second, it was hydrogen peroxide, right? So again, similar studies were done. The left side of the leaf vein had the sensor and the right side had the control. And if you look at these maps, if you see the red in the map represents high concentration, high fluorescence, and the right part, and when it goes to blue, it's low fluorescence. So with time, the H2O2 is being released. There is an increase in H2O2 due to this wound. That's how the plant is responding. And over time, you can see the fluorescence going down, right? So it's going down, and then again, it's responding back to its pristine state. So it's going down because this carbon nanotube, when it interfaces with H2O2, it has been designed in a way that its fluorescence is going to quench. which is why you see at T1 to 4 minutes, it's going from the red zone to the yellow zone. So the fluorescence is actually going down. And then again, it's going back to the normal states. So these sort of plant nanosensors can help you real-time detect changes in plant stress molecules. This is a non-invasive manner. You can interface the sensors in various plant parts. Number of sensory materials have been utilized. So you have polymer cap nanoparticles, you have silver and gold nanoparticles. The reason why silver and gold, particularly gold, is because there is a pretty interesting surface phenomenon in gold called surface plasmon resonance. And researchers have used this phenomena to ensure that when gold detects a particular stress molecule, its surface plasmon resonance changes. So you can identify the change in the output radiation, in the output signal, you can identify as being indicative of a particular stress molecule being released. And interestingly, this has been applied to a number of different plants. So leafy vegetables, tomatoes, spinach, strawberries, these nanosensors have been utilized to identify stress release in these plants due to abiotic stress factors. So the strategic advantages is, again, in situ monitoring of endogenous biochemicals. These are present in very small concentration. You can integrate the data that you get with real-time optoelectronic or smartphone-based monitoring systems, and you can get a thorough investigation into stress response pathways and mechanisms. Going into how we can use nanoscale materials for genetic engineering, the strategy here is if you have a nanoparticle conjugated biomolecular complex, so you have a nanocarrier that carries a particular DNA, RNA, or protein, and you can transform that into the plant, and in the plant, the DNA or RNA is expressed, and it can either lead to certain gene expression, or it can silence a particular gene, that is, do the opposite of expressing the gene. It can silence the particular gene, thereby imparting stress tolerance, pest resistance, and genetic engineering. But then what is the uniqueness of nanoparticles? So in general, interfacing DNA or RNA into the plant is difficult because the plant cell wall is a predominant barrier, right? So that is the one of the most important, one of the most problematic things in genetic delivery. In addition, this process is called genetic transformation. The transformation efficiency is low. There can be tissue damage, there can be unavoidable DNA integration. So to remove these barriers and to make sure that the integration is more specific, more tunable, we use nanocarriers. And a number of studies have been done in this regard. So the first one being gold nanoparticles were used to deliver small RNA molecules. Now, why small RNA molecules or siRNA? So siRNA in this study was found to protect the plant against bacterial infection. So the siRNA, it silences a specific gene that improves the plant resistance. And if you look at the figures, that's what is happening. So there are the authors have tested 4 systems. The first one is the control. Second one is only the gold nanoparticles. The third one in orange is the gold nanoparticles containing this gene, this siRNA. And the fourth one is just the plasmid in itself. And if you see the maximum protection or maximum efficiency is being done by the gold nanoparticles that contain this smaller siRNA, and that improves the bacterial resistance to Arabidopsis. So Arabidopsis is the target plant here, and in the left side, they are measuring the chlorophyll content, and in the right side, they're measuring the bacterial growth. And naturally, this system shows the lowest bacterial growth. In the next study, similar concept. In this case, the vehicle or the vector is not gold, but it's carbon nanotubes. So carbon nanotubes that are carrying genetic material for gene expression in plants. This is a very seminal study because this study showed how you can use high aspect ratio nanoparticles to enable functional genetic material delivery in plants. And these materials enter the plant by diffusion, right? So you have basically prevented the roadblocks encountered due to the cell wall being a barrier, or due to unavoidable DNA integration, or due to low transformational efficiencies. And they tested this system in number of four different plants. And you can see in all four plants, this gene has been successfully inserted, and it has shown a particular signal in all four plants. So the main point is the number of carrier matrices that are available that can help deliver your biomolecular cargo, DNA, RNA, protein into the plant. And it has been applied in a number of different plants, corn, maize, cotton, tomato, and so on. And it can help in effectively delivering the active cargo inside the plant. So the two main strategic advantages are targeted and effective delivery of cargoes. And once you have a particular cargo in the plant, it upon expression or interfacing in the plant's biomolecular system, it can impart stress tolerance, it can impart pest resistance, and so on. And you can leverage nanomaterial chemistry, you can leverage nanomaterial choice of vector selection, size, to overcome these transport barriers that are commonly faced when delivering active biomolecular cargo into the plant. Lastly, seed resilience. And the main focus here is how can we protect the seed from adverse environmental and climate related shocks and stressors, right? And number of studies have been done here, and I've presented two mechanisms. The first mechanism is, imagine the seed being in a coat that always provides it nutrients, right? So you have a coat that provides it a slow release source of nutrient over time, which enhances the germination of the seed. Or you could have hydrogel based coatings that provide the seed adequate moisture and improve shelf life. So if you have areas which are under drought, or which face a lot of heat risks, these mechanisms can help enhance the productivity, enhance the protection of the seed. It can help in the seed maintaining its conditions over time, leading to germination. And a lot of interesting studies have been done in this space. The first one of my favorites, is a double layer coating was a seed coating was prepared, right? So this provided moisture, but it also provided a particular species of bacteria called rhizobacteria. Now, rhizobacteria are very important bacteria which are present in the root nodules of plant, and they help in fixing nitrogen. So the authors said, if we could have a protective layer or a bilayer that not only provides an enabling moisture environment to the seed, but also specifically provides an environment rich in rhizobacteria, then it can enhance the germination of the seed over time. And they tried this in drought impacted areas. And what you can see is, in the coated seeds, clearly the plant growth is much more compared to the uncoated seeds. You can see the extent of roots that have been formed in the coated seeds. They are much more than the extent of roots in uncoated seeds. The next study is about a particular coating which contains zinc and hydroxyapatite. Again, it's a coating that provides phosphate and zinc micronutrient and urea to maize seeds. And if you look at the treatments on the treatment graph on the left, you'll see that the brown bar is the highest. It is statistically significant than the control, and it has higher germination percentage. Now, of course, in this study, often you'll not get enough effectiveness. Like you see here, that the zinc hydroxyapatite has almost the same effectiveness as as the coating itself. But again, this would have a much greater impact in areas or in regions that are facing climate related risks, or if you had to preserve the seed for a longer period of time. So again, a lot of coating materials have been involved. There is a lot of work being done right now on silk as a coating material for seeds, but other materials include metal oxide nanoparticles or electrospun nanofibers, hydrogels, polymers, and so on. And with a number of different plants like beans and broccoli and maize and so on, corn, whose seeds have been tested and tried using these coating materials, and effectiveness has been tried through investigations on shelf life, on packaging, and identifying how the plant grows over time when placed in mock climate risk conditions. So this strategic advantage here is you can create these localized enabling conditions for seed growth, for storage, for germination, and you can also, because you're working with the seed, once the seed is in the soil, there is a potential of positively conditioning the surrounding soil, promoting favorable germination conditions. And what do I mean by conditioning? Like we saw in the previous example, there was a localized concentration of rhizobacteria that was created, which led to increased root nodule formation and increased nitrogen fixation. So to boil this all down, like we have looked into a number of different applications. We have seen applications across the life cycle, across various regions of the plant. To boil this down, what are the environmental implications? Well, one of them is when you're applying these materials to plants, they help you prevent excess material consumption. They help in targeted delivery of nutrients, and they minimize environmental pollution. What is it for the soil? Well, you drive localized soil ecological and microbiological enrichment because of the slow inactive ingredient release, which when there is more diversity in the soil, it can drive a favorable growth environment for the crops. And what is it for the plant? Well, the plant gets more stress tolerance from climate adverse impacts. There is a reduced perishability of seeds. There is an enabling environment for the seeds to improve germination, for enabling environment to improve germination in adverse environmental conditions. And there is a potential for monitoring this data real time and sharing this data across different platforms and precision agricultural technologies and so on. Which then brings us to a question. If this technology is so interesting, it has so effectiveness, what pillars can guide that sustainable implementation of this technology? And I've organized this into 4 main pillars across health and safety, policy, materials design, and stakeholder engagement and adoption. And we are going to go into each of these pillars with the main questions being, for health and safety, what is the ecosystem and human health risk assessment, measurement, and protocols that are needed? For policy, what can the current policy landscape tell us about nanoscale-based technologies in agriculture, and what policy drivers are needed for further adoption? In case of materials design, how can we identify components of sustainable materials design, ingredients, recipes, chemistries, and how can we use metrics, well-known metrics like LCS and embodied energy calculations, to ensure sustainability is driven in materials design? And lastly, stakeholder engagement. Are we involving the right people at various stages of adoption of this technology to make sure that everyone is on board, to make sure that effective stakeholder output and input is both incorporated when these nanotechnologies are being applied in the field and while measuring the progress towards particular SDGs. Now, when I go into health and safety, there are some really good frameworks that have been created. So one of them is the One Health approach, right? So the One Health approach basically looks into 2 pillars. It looks into conceptualizing and planning or executing. Now, when I look into conceptualizing, it's basically identifying during the application of this technology, what are the environmental releases and human exposure and food health considerations that need to be incorporated, right? And how can we, in addition to the science, how can we integrate the consumer behavior, the market factors, the food trends, and so on? So this is the conceptualization that will guide the planning or execution of identifying health-related impacts. So what is the extensiveness of collaboration needed between chemists, regulators, and ecotoxicologists? Whenever we do health-based risk assessment, it needs number of characterization and QA/QC methods. How can we harmonize these across the different nano-based technologies in agriculture? And how can we integrate the data streams? Again, multiple data streams of toxicity, materials application, crops, and so on, to synthesize what is the monitoring and evaluation of health-related risks and risk impacts, and how can we ensure agronomist impacts and inputs are considered. So the One Health impact, the One Health approach conceptualizes, frames the question, and then develops a planning and execution method to identify how these questions can be addressed across different stakeholders. The other goes more towards measurement and monitoring, which are characterizing exposure and material investigation in biologically relevant fluids. And again, there are two steps. The first step being the initial determination of the nanoscale product meeting the regulatory definition of whether it is indeed a nanomaterial and if there is any additional assessment that is required. So for a bulk material, the characterization exposure would be done differently in a number of ways than it would be for a nanoscale material, because the toxicity and response of the nanoscale material and the host nanoscale material response would be very different compared to that of the bulk scale material. And the characterizations here would be along the chemistry of the active ingredient, the nanocarrier matrix, identifying material flows, and monitoring bioaccumulation, exposure, and tissue assay studies. The sustainability implications being, is the nanoscale formulation device or a formulation safe for human health? Because you're going to include it in the plant, and the plant is going to, the plant is going to be used, it's going to be either consumed. So is it safe for human health? Is the design and application considering different stakeholders like toxicologists and agronomists and so on? And are different data sources harmonized across this risk assessment and management method to facilitate decision making? In case of materials design, a study that was performed, they found that compared to traditional fertilizers, nanoscale amendments do not actually posit a sustainable alternative. So if you they measured and monitored the embodied energy of these different nanoscale materials, so that's the orange data points, and the blue bar over here is the embodied energy of conventional nitrogen fertilizers. So you can see that the embodied energy of these nanoscale applications is much more. However, if you have a more combinatorial approach, so you replace the bulk fertilizer with the nanoscale material at various proportions, then you can achieve a more sustainable application. You can achieve a more sustainable materials-based design approach, and it will lead to a lower embodied energy per volume of soil. Because the whole purpose is here that the material selection, the dose, the way you are mixing the nanoscale material with the bulk application, that can affect the embodied energy of the application. And the whole purpose towards sustainable application is to make sure the embodied energy is as low as possible. Now, they also tested different materials for as seed coatings and for applications in foliar leaf-based applications, and they found out that if I compare the yield, if we compare the increase in yield to the embodied energy, the authors found out that the materials have a large, they are spread across a large matrix, and you would naturally consider those materials that have a high increase in yield and low embodied energy. Now, this study is a very interesting study because it tells you that it's very important to characterize the embodied energy, the life cycle inputs of these materials to identify which material is good for which purpose. So, for example, in this figure is not well visible, but carbon nanotubes, yes, they have embodied energy and they are good. They provide a good increase in yield, so they are a good selection. They're a good candidate for seeds. But if you look into plants, maybe not. Their increase in yield is not enough, but they do have high embodied energy. So this sort of decision making can help you identify for which plant, based on the target application, be it as a seed coating or be it as a nanosensor or be it as a agrochemical, a nanopesticide or nanoagrochemical, what is the material that you want to use and what is going to govern the usage and application of that particular material. In addition, there is a need to better get LCA data and GHG emissions data during formulation use and post-use strategies. So this study focuses on embodied energy, but it also lays the case of how these indicators can help towards identifying more informed application, benchmarking, and assessment. I also looked at the policy landscape. So what is the policy landscape surrounding nanoagriculture, right? And it's very important because if we want to increase the application of this technology going forward, then we really need to know how it can be governed and how various stakeholders are they are well aware of how to use how to use these technologies going forward. So I looked at a very broad snapshot, and in the EU, the biocidal products regulation is currently the only regulation that specifically addresses nanomaterials through various methods like labeling, packaging, risk assessment, and so on. And they have candidates that you can use in agricultural applications. Other frameworks that can be very helpful. So for example, the REACH framework. So the REACH framework provides indicators of test guidelines, safety testing, it provides how the various risk assessment and measurement methods based on nanoparticle physicochemical properties, human health exposures, toxicokinetics, and so on. And also the EFSA, the European Food Safety Authority. It provides guidance on risk assessment of nanomaterials to be applied in the food chain, and how conventional risk assessment should be complemented with nanospecific considerations. So these European guidelines can provide helpful indicators in testing, risk assessment, and monitoring of nano-based technologies in food products and in general in agriculture. If I look at India, India has certain policies that ensure the safety and efficacy of these innovations to promote their use in agriculture. And this guidance will help in the development of regulatory processes towards commercialization. So how can nano-based sensors or nanopesticides be commercialized, and what are the guidelines that are required to do so? In Canada, there is work ongoing on nano-based methods and technologies. There is limited regulation in the food space, but there Health Canada has a lot of attention on risk and benefit screening of nano-based health and food products. This includes specifically manufacturers to engage in product safety assessments, and there are certain parameters that need to be screened, or that manufacturers are advised to screen on, such as nanomaterial use and methods, manufacturing, materials characterization, and so on. Which tells us what are some of the enablers of robust policymaking in this space. So from a legislation standpoint, there is a need to include and understand the definition of nanoagrochemicals or nanopesticides and materials considerations. There is a need of standardization in protocols. How can you formulate these materials? How am I using them in the field? What is the risk assessment? How is the post-use management going to be considered? And human. There's also the need of financial and human resource capacity to facilitate regulatory cross-agency collaboration and facilitate R&D efforts, and make sure that the intended economic, social, and environmental benefits are being realized, and identify the role of emerging technologies to establish scientific benchmarks. That is from a regulatory or legislation-based standpoint. The next thing is a stakeholder engagement standpoint. So how can we enhance public awareness on the environmental fate, disposal, and recycling of nano-enabled products? What training and capacity building needs to be ensured? How can different social and cultural contexts be incorporated? And lastly, monitoring and evolution. Enhanced risk screening and assessment, guidelines for materials characterization, toxicity studies, and facilitation of nanoagrochemical and pesticide-specific databases. Finally, what is the extent of stakeholder engagement that is needed? And the reason I place this slide is because I realize whenever we want sustainability we want a particular technology to be applied sustainably, it really needs feedback from various stakeholders. And the bottommost pillar, or the most important pillar, is the collaboration between scientists, agronomists, and farmers, because they are the primary users and generators of this data. So there is a real need of translating and sharing knowledge between these three pools of these three pools. Once that knowledge is shared between these three pools, this can inform local and regional government planning. It can lead to evidence-based policy making. It can identify integration with local climate resilient agricultural infrastructure and climate smart agriculture priorities. And it can drive at a higher level, it can drive the creation of standards using local and regional knowledge. It can promote global cooperation and coordination. It can provide technical and public-private finance. And this is a very rough framework where I believe we need the stakeholder engagement to drive the application of these technologies in agriculture in a sustainable manner. So to conclude, nano-based applications have substantial potential. There is a lot of positive benefits that one can realize from this technology, but then we really need to take a step back and understand where do we need to work to enable policies? Where do we need stakeholder input? How can we really ensure that ground actors, farmers, agronomists have their decisions and their inputs are being considered, and where technology readiness is being needed, is required. So for different nano-based applications, where is a need of R&D to be, where do we need more R&D? Where do we need finance to scale up from laboratory to field trials? And how can we communicate the value of this technology, the effectiveness, but also the drawbacks and the need for more work to different stakeholders? And lastly, how can we have better data acquisition and monitoring methods to make sure that the methods of formulation, application, and post-use are actually sustainable, and there is robust data to suggest that. Thank you very much.